Hello students, welcome to the channel. So today in this video we are going to discuss some questions and with their brief explanations. So here we have a first question is that nephelometry and turbidimetry is based on the principle of, and here we have option as A, light absorption, B, light emission, C, light scattering, D, or all of the above. So the correct answer is light scattering. So we can now understand with the help of explanation that what is the principle of nephelometry and turbidimetry. So the principle of nephelometry and turbidimetry is based on the scattering or absorption of light by solid or colloidal particles suspended in solution, because when light is passed through the solution, then the part of incident radiant energy is dissipated by absorption, reflection and reaction, while remainder is transmitted. Students, when just pass a light through the sample, some light is actually absorbed and some is transmitted. So that transmitted or scattered light is the measurement of intensity. So like we just see in this line, that measurement of the intensity of transmitted light means that light which is transmitted after the absorption by the solid particle is measured as a function of the concentration of dispersed phase, and this becomes the basis of turbidimetric analysis. Now we can also understand with the help of diagram and further coming in case of here we have a turbidimetry diagram, that there is a light source and here we have a lens and samples to be analyzed. So again we have a lens. So after, when the light source is giving a light and it passes through the lens and passes from the solution, then there will be, it also goes through the lens, and the light which is scattered or transmitted is detected by the detector. And in case of nephelometry, there is a light source and a lens and the sample which is to be analyzed, and the light which is scattered after passing from the solution is detected by the detector. So here it's just all about the information about the turbidimetry and nephelometry. So try to memorize the principle of these because it is very important. So the next question is causative organism for plague is, and here we have options are A, Coaxella burnetii, B, Leishmania, C, Yersinia pestis, D, Flavivirus fibricus. So the correct answer is Yersinia pestis. So just to discuss about the plague or the causative organism is, let's discuss about the plague, that it is an infectious disease caused by the bacterium Yersinia pestis that affects the human and other mammals. Actually, the plague get it humans usually get plague after being bitten by the rodent flea that is carrying a plague bacterium, or by handling them. And the symptoms include the fever, weakness and headache. So it's all about the plague. The next question is, in budget 2019 and 20, the government set the target to distribute LPG connections under the Ujjwala Yojana. So the options are A, 6 crore, B, 10 crore, C, 5 crore, or D, 8 crore. So the correct option is that the government set the target to distribute around 8 crore LPG connection under the Ujjwal Yojana. So let's discuss in brief that the government has set the target to distribute 8 crore LPG connection or cylinders to the household under the Ujjwal scheme. Students try to learn both of them, that what is the scheme name and how many connections distribute under the Ujjwal scheme. So far government has has distributed around 6 crore cylinders under this scheme. So the next question is, which one of the following is a dopamine agonist? And here we have options are A, entacapone, B, phenelzine, C, salicylic, D, bromocriptine. So the question asks that which one of the following is dopamine agonist, and the correct answer according to this is bromocriptine. So now we just discuss about the from the classification part that entacapone is a COMT inhibitor, phenelzine is irreversible MAO inhibitor, salicylic is MAO inhibitor, bromocriptine is dopamine agonist, which is asked in the question part. So don't confuse in classification. Try to revise on time, and because it is very helpful. And in previous year question paper, the various question asked from the classification part. So students, make your strong point in classification, so you don't need to get confused in exams before after just attempt the questions. So the next question is, which one of the antibiotic can cause visual damage? And the options here we have A, quinidochlor, chloroquine, C, tetracycline, and option D is chloramphenicol. So again the question asks that which one of the following cause visual damage? So the correct answer is chloroquine. So now we just understand with the help of this that quinidochlor is responsible for blindness in children, nephropathy syndrome called subacute myelooptic neuropathy, and goiter, where chloroquine, which is asked in the question, is having visual damage as a major side effect, and loss of hearing and myopathy. And it is about tetracycline, that kidney and liver damage, and the discoloration of teeth is the major adverse effect of the tetracycline. Students, try to note down in your copy right now, because many a times this question asked in NIPER exam or pharmacist exam or GPAT exam, that which is the major side effect of the tetracycline. The correct answer is discoloration of teeth. And now we just talk about chloramphenicol, and its major side effect is gray baby syndrome and BMD and hypersensitivity reaction. So it's all about the major side effects. So students, try to learn them with very ease. So the next question is, who is the chairman of ISRO? And the options are A, Shailesh Nayak, B, Kailasa Vadivosevan, A.S. Kiran Kumar, K. Radhakrishnan, and the option is B correct, and K. Sivan is the chairman of ISRO. So the next question is, SDS page is known as, and here we have options are A, sodium disulfide polyacrylamide gel electrophoresis. Next option is sodium dodecyl sulfonate polyacrylamide gel electrophoresis. C option is sulfur disulfoxide particle average grade electrophoresis, or D, none of the above. So the correct option is that SDS page is known as sodium dodecyl sulfonate polyacrylamide gel electrophoresis. So let's discuss in brief that SDS page is sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is an analytical method used in biochemistry for the separation of charged molecules in mixture by their molecular masses. Students, in this, there will be a separation of charged molecule based on their molecular masses in the presence of electric field. It uses sodium dodecyl sulfate. Sodium dodecyl sulfate used by this molecules to help in identification and isolation of protein molecules. Let's discuss with the help of a diagram that there will be a gel cassette, and negative electrode chamber, and there will be anode buffer, cathode buffer, and here we are just we are add the sample, and under the electric field, suitable electric field, according to the molecular masses of the molecules, the separation will occur, and it is helpful for the identification and isolation of the protein molecules. Protein, that we can observe that here there is a line written that separate protein bands. So students, it is all about the SDS page. So next question is antibody which is responsible for agglutination of blood is, and here we have options are IgA, IgD, IgE, or IgM. So the correct answer is D, IgM. So now we just talk about the antibodies that IgA present in saliva and tear, and the main function is to protection to mucous membrane. Sometimes the question also may arise that what is the function of IgA. And the correct answer is to protect the mucous membrane and cavity from infection. And what about IgD? IgD actually present in serum and lymphocytes, which control antigen stimulation of beta cells and fetal antigen receptor. And IgE is combat parasite disease, causes allergy, drug sensitivity, and hypersensitivity. And what about IgM? IgM is present in serum and lymph and agglutination of blood. Then the function of this is protection against infections caused by bacteria. Students try to learn that where the antigens are present and what are the major functions of these. So the here you can also learn that here you can also learn in the with the help of this that A, D, E, M, ADEME. You can learn with the help of these mnemonics in series wise. So here there is no chance to forget about the where they are present and the main functions. So it's all about the antigen and antibodies. Sorry. So the next question is, in biopharmaceutical classification, the class 2 drugs are with, and here we have options are option A, low solubility and high permeability, option B, high solubility and low permeability, option C, low solubility and low permeability, option D, high solubility and high permeability, and the correct answer is low solubility and high permeability. So now we can understand with the help of this that according to the biopharmaceutical classification, there are 4 classes: class first, class 2, class 3, and class 4. According to the class first, there will be high solubility and high permeability. According to the class 2, there will be low solubility and high permeability, which is asked in the question. And according to the class 3, there will be high solubility and low permeability. And at last, the class 4, there will be low solubility and low permeability. So the correct answer is class 2, low solubility and high permeability. So now next question is, which of the anti-cancer is having anti-inflammatory activity? So the options here we are having the A, methotrexate, B, cyclophosphamide, C, tacrolimus, and D, all of the above. So the correct answer is methotrexate. So according to the explanation part, that methotrexate inhibit dihydrofolate reductase and is used in the cancer, uh, and it is also used for the treatment of rheumatoid arthritis because it inhibit proliferation of the lymphocytes and other cells responsible for inflammation in the joints, whereas cyclophosphamide is a nitrogen mustard which is used in cancers, and tacrolimus is a calcineurin inhibitor and is used in as immunosuppressant and also used in the rheumatoid arthritis. So the correct answer, hence the methotrexate is the correct answer. So the next question is, in case of uncompetitive inhibitors, the values of both Michaelis-Menten constant, where that is Km and Vmax are, and here we have options are Km increases, Vmax unaffected. B option is Km unaffected, Vmax increases, and C option says Km unaffected, Vmax increases. D option is both Km and Vmax decreases. So answer is both Km and Vmax decreases. So now just understand by this explanation that in case of competitive inhibition, that Km is increased and Vmax is constant, and in non-competitive inhibition Km is constant and Vmax is decrease, and uncompetitive inhibition case both Km and Vmax decrease. Students, try to just revise these things, don't get confused in the case of competitive, non-competitive and uncompetitive. Try to learn and get revised on time, so there will be no chance to get confused in these types of cases. So the next question is, aspirin is, option A is selective COX inhibitor, option B is non-selective COX inhibitor, option C is reversible LOX stimulator, option D is a reversible LOX stimulator. So the correct answer is non-selective COX inhibitor. Here we have a explanation, and here we have a classification of this, that we can understand by that there are non-selective COX inhibitor, salicylates, which is asked in the question that what are actually aspirin is. Aspirin comes under the category of non-selective COX inhibitor, and here we have a preferential COX inhibitor 2 inhibitor and selective COX2 inhibitors and analgesics, antipyretics. Students, try to revise these classification on time. So next question is, which one of the following vitamin act as a coenzyme and carboxylase enzyme? And the correct options are, A is pantothenic acid, option B is biotin, and option C is niacin, and option D is cobalamin. And the correct answer is biotin. Biotin is the vitamin which acts as a coenzyme for carboxylase enzyme. So let's discuss in a brief. Here we have vitamins and with their coenzymes. So in case of thiamine, the coenzyme is thiamine pyrophosphate TTP, and in case of pyridoxine, the coenzyme is pyridoxyl phosphate. In case of nicotinic acid, the coenzyme is nicotinamide adenine dinucleotide, that is NAD plus. And in case of pantothenic acid, the coenzyme is coenzyme A, and in case of biotin, the coenzyme is carboxylase, which is asked in the question. And the last question is, who is known as the father of plastic surgery? And here we have options are Charak, G.D. Nade, C option is Sushruta, and D option is none of the above. So Sushruta is the father of plastic surgery. Let's discuss in brief explanation part that Sushruta is the founding father of surgery and plastic surgery. He had also written Sushruta Samhita, an outstanding treatise in Indian medical literature, and describes the ancient tradition of surgery in India. It's all about the questions. Thank you.